potential is the movement of an electrical impulse throughout the axon terminal. This occurs when depolarization goes to a particular value called a threshold. A strong neurostimulation creates a change in electrical charge, causing an action potential to be produced. This happens over a series of 5 steps: the resting state, depolarization, the rising phase of the action potential, the falling phase of the action potential, and the undershoot. The action potential or impulse occurs in a region called the axon, as being shown on the board right now.
The first phase of this process is the resting phase, in which the Na and K channels are closed and the ungated channels maintain resting potential. In the second phase, depolarization, a stimulus causes the sodium ion gated channels to open. The membrane is depolarized through the inflow of sodium ions. Once the depolarization reaches threshold, an action potential will be triggered. In the third phase, the rising phase of the action potential, the inside of the membrane is relatively positive compared to the outside due to the influx of sodium ions. The fourth phase is the falling phase of the action potential. In this, the sodium channels are inactivated, which block the sodium flow. On the other hand, potassium channels open up, which increases the negativity inside of the membrane. The fifth phase is called undershoot, where the membrane returns to resting potential. To summarize, the impulse starts with the membrane in resting potential as seen in step 1. In step 2, the impulse depolarizes the membrane potential which increases to a point called the threshold potential. After reaching the threshold potential, an action potential is triggered, which causes the membrane potential to increase significantly, as seen in step 3. This is because most sodium channels are open and the inside is much more positive than the outside. In step 4, the inactivation or blocking of Na channels and the opening of K plus channels causes the membrane potential to fall as the inside of the cell becomes more negative. Finally, in step 5, the membrane potential falls below the resting potential because some potassium channels are still open. When the potassium channels are closed, the Na channels are unblocked, though still closed, and the membrane returns to its resting potential.
Transmitted across the synapse. Signals in nerve cells refer to the action potentials which flow along axons, which is the middle length of the neuron drawn here. To clarify, the action potentials themselves are not transmitted, but information is transmitted, and this transmission occurs at the synapses. Synapses are junctions between two nerve cells consisting of a minute gap across which impulses pass by the diffusion of a neurotransmitter. Being drawn here are the two axons, which are vital to the transfer of the information. As you can see, the axon spreads out its fingers or terminal branches. The end of these branches are terminal buttons like the one shown on the left of this image, from which the neurotransmitters are emitted or reuptaken based on the situation, such as demonstrated by the red arrows, which are going out or into the presynaptic vesicle. The terminal branches and the dendrites of the postsynaptic neuron are separated by a space called a synaptic cleft. This description is being drawn here, and the box represents the synaptic cleft, which is where neurotransmitters are pulsed out of the presynaptic neuron into the receptors of the ligand-gated ion channels in the postsynaptic neuron. The opening of these channels floods sodium and potassium ions into the adjacent cell and triggers its membrane potential. Soon, a chain reaction is created as many more neural connections similar to the one shown on the board are made, and various electrical impulses travel through a web of neurons. So, to sum up, first, an action potential or electrical signal is generated near the cell body portion of an axon when depolarization reaches the threshold. Think of a cell phone. The electrical signals in our phones occur due to the flow of electrons. Similarly, the electrical signals of our nerve cells are a result of ions moving across the neural membrane. The signal produced is sent across the axon until it reaches the terminal branches. At terminal branches, the presynaptic neuron sends its neurotransmitters, which are stored in vesicles within the terminal button, across the chemical synapses through exocytosis and into the synaptic cleft. Neurotransmitter release triggers the opening of the ligand-gated ion channels on the tips of the dendrites of the postsynaptic neuron. The release triggers a chemical membrane potential in the postsynaptic neuron, inducing an axon potential and starting the process all over again. Ultimately, through cell communication, signals are not only created but transferred, neuron to neuron, contributing to our efficiency as a higher-order mammal.